In today's episode, we'll be going through nucleotides and nucleic acids exam questions. How I'm going to approach this is that I'm going to show you from start to finish how I'm thinking about it, how I'm going to approach it, and the structure of my answers, and then we're going to compare it to the mark scheme. So, let's begin. Suggest a suitable substance that the student could use to release DNA and explain why it should work. This topic, of course, is an AS topic. And when it comes to AS, the answers to the questions are much easier to predict, I guess, and just the mark scheme is so much more simpler than if it was like an A2 topic and you're like going towards, exam season and stuff in year 13, and you look back on AS papers, you realise that is so ridiculous. Like, it's so easy after doing A2, okay? Because you realise it's a whole different ballgame. The actual A-level papers compared to the AS papers. So just trust that all the AS topics are so easy in terms of the mark scheme answers. Just know the content. But A2, you need to know the content and also know how to apply it. A suitable substance that can release DNA. Okay, detergent. Why? Because I know the mark scheme loves detergent for exam questions. And why it will work? Well, detergent. From my note, I remember that it tracks the phospholipid molecules and the water molecules. So if it's attracting them, then it will break up the phospholipid bilayer and thus it will release the DNA. Next questions. What type of chemical reaction takes place when two nucleotides in a single polynucleotide strand are joined together? Easy, condensation. Name the chemical released when the bond is formed between two nucleotides. Again, very easy. Water. Content-based answer again. Now, this one. Describe how these two chains are held together. DNA molecule contains two polynucleotide chains. Essentially, describe the structure of DNA. So, we know that there are hydrogen bonds. So there's hydrogen bonds, phosphodiester bonds, which form the backbone. We have purines to pyrimidines, so A to T and C to G. A to T, 2 hydrogen bonds, C to G, 3 hydrogen bonds, and that's more than 3 marks. So let's write that down. Okay, let's check the answers for this whole page. This one. Right, so we said about detergent, we said about it tracks the phospholipid molecules and it breaks up the plasma membrane. What I should have actually said is about the emulsifier. That's such an easy term you can just remember and put in whenever you're talking about detergents. So that's a note I would make in my notes if I was doing this A-level again. Right, and the next one, condensation water, very simple. This one, phosphodiester bonds. The backbone, hydrogen bonds. Yep. Easy. As I said before, AS topics are just content. Literally just content-based questions. So yeah, now it is your go. Have a go at some of these questions and let me know how you do.